Hello, in this video we will look at the reabsorption in nephron. So the reabsorption in the nephron after the filtrate are present in the nephron, which that is through glomerulus pressure will create the filtration. So the kidney, the functional unit of the filtration is the nephron of the kidney. So in this way, here is the 180 liter fluid circulation per day, and this reabsorption can be by active transport, and this reabsorption and secretion will be occur, and diffusion, osmosis facilitate diffusion and import, as well as the active transport basically. This all things is required for the reabsorption of the waste for the useful material. So in this way, here is the reabsorbed 99% and 1% excrete during one cycle of blood circulation into the kidney. So in this way, here is the nephron. This nephron is contain proximal convoluted tubule, loop of Henle, and distal convoluted tubule and collecting duct. Collecting duct contain a distal collecting duct and proximal collecting duct. So in this way, here is you can see that is the glomerulus. This glomerulus contain afferent arteriole, which that is lead to move toward the glomerulus, while the efferent arteriole, which that is used for the reabsorption of the filtrate. So the filtrate mean the after the excretion of the waste into the nephron tubule, so that is known as the filtrate. And here is the basically vasa recta form, this efferent arteriole, which that is surrounded to the loop of Henle. So in this way, after the movement of the ion and compound and molecule, which that can be sodium, potassium, calcium, magnesium, water, uric acid, urea, glucose, amino acid, protein, vitamin, and lactate. This can be reabsorbed and in the proximal convoluted tubule through a facilitated diffusion or direct diffusion or the active transport. So in this way, basically, is the urea is also secrete, and the proton as well as the amine group and creatine or some drugs can be secrete from the efferent arteriole more. So in this way, the secretion of the urea also through the efferent arteriole and drugs and proton and amine. So in this way, here is you can see in this diagram to understand the loop of Henle, which that is reabsorb the water while the secrete the urea through efferent arteriole into the loop of Henle. So the descending loop of Henle is the thin, and the ascending loop of Henle contain a thick structure, which that is the epithelial thickness is more. So in this way, the beginning of the distal convoluted tubule, the import transport will be occur, which that means the co-transportation as well as the sim transport. Mainly the sodium, potassium, and chloride collectively move out into that efferent arteriole. So that is known as the reabsorption. So after this, here's the also the distal end of the collecting tubule. Basically, the sodium chloride and hydrogen bicarbonate and water is more absorbed. And collecting duct at the proximal course of the collecting duct, which that is secrete the proton, potassium, and amine, while the distal end of the proximal tubule collecting tubule distal collecting tubule, which that's contain aquaporin channel. This aquaporin channel will reabsorb the water, and this urea is absorbed secrete, while the aquaporin will reabsorb. So in this way, the aldosterone, which that lead to produce the aquaporin channel through a release through adrenal gland, while the pituitary gland, the neurohypophysis will secrete the antidiuretic hormone. This antidiuretic hormone will stimulate the aldosterone to create the more aquaporin type 2 channel for reabsorption of the water. So this is known as the facilitated diffusion of the water into the efferent arteriole. So this is the vasa recta. Vasa recta basically is the afferent arteriole make a surrounding to the loop of Henle for reabsorption of water and some ions. So in this way, the 95% water will be excreted even. So on the other hand, important thing is that if the diabetic person which that is increase the blood glucose level will excrete the blood glucose level for the maintenance of the glucose. So while the normal person will not do that. So here is the proximal convoluted tubule we will discuss first. So this is the interstitial space and between the uh, uh, epithelial cell and between the epithelial cell and blood stream, the blood vessel. So this is the interstitial space and this is the epithelial cell and here is the lumen. So here is the proton will be pump out and the sodium molecule will be in move one molecule, one ion. In the case of the inside movement into the efferent arteriole, so the ATP is required for the sodium 1 move inside for the efflux of the potassium. So in this way, here is the proton basically will move out to the chlorine and sodium co-transportation to move inside. So on the case of the basically is the proton will move out for the one phosphate group phosphate to bind with the sodium to move in and their diffusion into the bloodstream through a facilitate. So in this way here the proton will move out to calcium will move inside with the co-transporter sodium. So this is the sodium is the co-transporter. It is also known as the symport, but it is not symport, it is co-transport. While the amino acid is also required the sodium and glucose is also required the sodium for same mechanism. So in this case, the magnesium will be reabsorbed directly into the blood vessel and water also in the proximal convoluted tubule. But in the proximal convoluted tubule, the pH balance in blood, so the sodium will move inside, and this in this way, the basically the hydrogen bicarbonate will be converted into the carbonic acid through carbonic anhydrase enzyme. So important to know that if the pH will be high into the lumen, so the carbonic acid will be formed. For decrease the pH, a more acidic. So in this way, the uh, if the pH will be low, so that will be dissociate and produce the water and carbon dioxide will be diffused directly into the epithelial cell and the water also. So in this way, in same mechanism are present in the epithelial cell as well as the blood. In this way, the acid-base balance in the blood as well as the collecting as well as into the lumen of the nephron. So let's begin to understand why, because the bladder remain the normal pH. So that is why the pH balance into the urine is so important. So the basic and acidity is due to the dissociation and association of the carbonic acid. So in this way here you can see that is next we will understand into the loop of Henle. So the loop of Henle contain a descending and ascending loop of Henle. Ascending is the thick and descending is the thin. So the water will be reabsorbed into the beginning of the descending loop of Henle. So after the reabsorption of the water, so basically the high concentration of sodium will be occur. So that means the concentration of the solute will be more as compared to the solvent as a water. So in this way, the movement of the molecule is the basically the concentration is the 1200 milliosmol per kg is the uptake of the water. So that is the diffusion rate, and in this way you can see that is the decrease the upward side. Why? Because the amount of water will be reabsorb into the ascending loop of Henle will be decreased, while the increase of the sodium reabsorption through active facilitate diffusion. So the active transport basically the ATP use. So in this way the water will be more into the ascending loop of Henle. So that means the interstitiums contain a basically 1200 milliosmol per kg, and that means the upward is the 200 milliosmol per kg. So it will be increased downside. So you can see, and the concentration of the sodium will be increased in the descending loop of Henle, which that is the passive diffusion will be occur, and this diffusion will move upward toward the ascending loop of Henle. So in this way, why? Because the decreasing of the sodium through active reabsorption to make a more dilution, and the more concentration of the solution will be more into the descending loop of Henle, but the ascending loop of Henle will be less. So in this way the counter current multiplier will be occur. In this way again and again the sodium reabsorb and the water will be diffused to move upward. So let's begin to understand the other thing is that the other thing is the distal convoluted tubule. So the distal mean the end of the nephron. So this is the distal convoluted tubule is contained basically is the lumen. Here is you can see, and this is the epithelial cell of the nephron. So this epithelial cell of the nephron basically is looking like this, and the sodium will move inside and the chloride will be moved also inside, is known as the symport transportation. So the chloride will be moving to the blood circulatory system, while the calcium and magnesium is directly diffused into the intercellular space, and in this way the water is also moving inside. So in this way the magnesium and calcium can be moved inside directly into the distal convoluted tubule and to move into the blood circulatory system, while the sodium will be exchanged by ATP and the potassium will be secreted. So next here we will discuss about the acid-base balance in the distal convoluted tubule. So that is the same mechanism basically is the acid-base balance due to the carbonic acid first, and there are other acids can be, but here is we can understand with the carbonic acid just. So in this way the lumen here and the carbonic hydrogen bicarbonate will be associated to form a carbonic acid, and this carbonic acid can be dissociated on the basis of acid base up down. So in this way the carbon dioxide will directly diffuse, and that's again that mechanism will be occur, and in this way the hydrogen bicarbonate will be moved or into the through a chloride co-transport. It is known as a symport transportation. So in this way the hydrogen bicarbonate can be associated with a proton to again form a carbonic acid on the basis of if the pH will be more, so the for the decreasing of the pH with carbonic acid will be formed. So here just we will understand about the distal end of the collecting duct. So this is the distal end of the collecting duct. The distal end of the collecting duct is basically the hydrogen bicarbonate will be associated with the proton and to form a carbonic acid, and it can be dissociated on the basis of the acid. If the acid will be more decreased, so the dissociation will be increase the basicity of the blood as the lumen. So in this way here is you can see in this diagram. So also produce the aquaporin 2 channel. This aquaporin 2 channel is stimulated by the aldosterone, and it is used for the more reabsorption of water, and it means that 95% water also still here, but the waste will be occur urea and uric acid and the potassium, etc. So thanks for watching.